Should I start? Okay. Okay, sure. Okay. Okay, thanks. So, hi, everyone. I'm going to talk today about accelerating biological sequence analysis via new algorithms and architectures. Particularly, we're going to focus on genomics and metagenomic sequences. And I will try to provide the necessary background that will be needed for this lecture. So it's okay if you don't have any prior background in these directions. Okay, a bit about me for people who do not me. So I'm a last year PhD candidate advised by Professor Onur Mutlu and in Safari Research Group at TCuARCH in the ETH department. And I also in the past have worked as a visiting researcher at Robust Systems Group at Stanford. And my research interests are generally in the field of computer architecture, with some focus on data-intensive health-related applications, particularly bioinformatics and the intersection with the storage system. You will see some of it today. And there are some links to contact me and to my website, and of course, I assume you know the group's website. So the agenda I have for today is as follows. I will give a brief overview of genomics and metagenomics, and we don't need to know anything about it in advance, not even what a camera is, because it will be covered. And why do we need to accelerate it? And why do we need algorithm architecture co-design for this acceleration effort, what it enables? I will give some very high-level examples of what can be potentially enabled. And also, I will dive a bit deeper into doing algorithm architecture co-design for these applications by going through some examples of storage-centric system design. And for each of the examples that I will give, I hope that we can cover some unique angles of doing hardware-software co-design for these domains. And I will also do some advertising for our new initiative, Architecture for Health, or ARC for Health, that we want to motivate in the broad community. So let's get started. Some brief overview of genomics and metagenomics. First, why are we going to talk about these? Because there are many important applications that are heavily impacted by these fields, such as uncovering and treating diseases that are linked to genomic variation, developing personalized medicine, which leads to much accurate results compared to other methods in many cases, detecting pathogens in the environment. So we have seen, unfortunately, what has happened in the previous pandemic, but if we are able to detect pathogens in a very fast way, in a proactive way constantly, then we can ensure that the next outbreak remains contained or remains in a scale of, let's say, epidemic, as opposed to turning into a global pandemic. So these are some things that we cannot, let's say, risk to repeat the history at that scale. So, and also, again, like rapid surveillance of disease outbreaks. So these applications and many, many more require us to understand the genomic information that is encoded in an organism's DNA. And to get these genomic information into digital data for us to be able to computationally analyze, we rely on these machines that are called sequencers. So they get the DNA molecules of organisms as input and give us these digital data as output so that we can computationally start analyzing it. But then the current sequencing technologies cannot sequence very long DNA molecules end to end. So what they typically do is that, so if you have a DNA molecule as an input and this sequencer machine, they extract the smaller fragments of the original DNA sequence known as reads. So then what we need to do in many genomic applications, in the first key step, is to do something called read mapping. So let's say I have these reads from some organism or some sample, let's say a human sample, and that's a reference genome, let's say like a relatively healthy human, so that's a reference. So we want to compare, do some read mapping to basically align the reads to some potential matching locations in this reference genome. And in the matching locations, with some expensive operation of alignment, find the degree of similarity and differences between them. And then later on in downstream steps, these similarities and differences can actually hint to some important biological or medical clues. And enable a lot more accurate diagnosis or let's say understanding of a complex environment compared to prior types of, like many other types of tests that are done. But then performing these alignment step is very difficult because we need to perform computationally expensive approximate string matching to amount for differences that can arise actually either due to sequencing errors or genetic variation. So in the end, what happens is that we have a lot of data to work with and a lot of expensive computation to perform. So if we have this high-level abstraction of the system with the storage system, main memory caches, and computation units, which can be CPUs or any accelerators, or I don't know, FPGAs or GPUs, what we need to do here then is to do a lot of expensive computation. So we have a computation overhead on a lot of data. So that needs to come all over. So we have also a lot of data movement overhead. So now let's take a look at metagenomics. What is that? So it's the study of genome sequences of diverse organisms within a shared environment, such as blood, ocean, soil. And it's quite important to be able to analyze such types of environments because currently, for example, we don't know more than 99.9, even more than that, organisms that are covered on the surface of Earth, and actually understanding it. Of course, like there are a lot of medical and biological benefits, but also there are a lot of environmental benefits as well, because we can understand what are the procedures that we have oxygen generated, or like the bacteria in the ocean that are responsible for that. And also recently, for example, people have shown discovery of plastic digesting bacteria. So you can see that the implication of such fields are actually huge. So since we do not know that what is in the sample in advance, what we actually need to do is to figure out what are the species that are present. And to do that, let's now consider some of these genomic sequences here. In high level, these three key steps are more or less what happens in many metagenomic analysis tools. So I'm going to abstract it away. Of course, there might be differences, but this is like, let's say, some high level way to look at it. So it's to preparation of input queries. So we would extract some smaller sequences of length k, which would be called k-mers, out of these input sequences, so that now we can, with these smaller chunks, we can start searching them. So the next step, of course, is to find what is present or absent in the sample. So we get these smaller subsequences and look them up in a large database containing information on many species. And these databases can be very large because depending on use cases, we might want to discover what's in the species and might not find it in like some usual databases. So that size can actually expand very large, to be very large. And after we figure out what's in the sample, then there are many other steps that can happen, but a common way to look at it is to find out the relative abundances of these species that we have identified in the sample. Okay, so that was kind of a high-level background that we need for genomics and metagenomics for this talk. And now I will talk about the need for accelerating it and some of the examples. So to generate these digital data from DNA molecules for us to analyze, we rely on these special purpose sequencing machines for data generation. And they have very high throughput, and their throughput has been actually increasing at a very fast rate. And their cost has been also dropping really quickly as well. So then relying on our conventional computing systems to analyze this volume of data leads to huge throughput mismatch. So we would be bottlenecked by computation if we rely on these general purpose computing systems. And some challenges would arise in terms of performance and scalability. Cost effectiveness, of course, like we can always scale up, add more and more processing units to just scale up what we have, but then leads to cost effectiveness issues and energy inefficiencies. And of course, a key challenge in genomics and metagenomic data is regarding privacy and security, which is not particularly what I will talk about in this talk, but it's also a very important challenge. So algorithm architecture co-design for accelerating metagenomics, I will try to motivate why that will be beneficial by talking about some of the high-level examples from our group and what they enabled. And after that, as I mentioned, we will go through some details by looking into some works in more detail. Okay, so you have seen this in this lecture, I assume, several times. So this is the expanded view of computer architecture. We have some algorithms to work with, going down to devices. In this talk, we will see how actually the algorithm, in some of the examples that in detail I will talk about, we will see how we fundamentally need to rethink our algorithmic trade-offs based on these lower layers of the system. Anyway, because of the importance of this domain, there's been a lot of effort from both industry and academia to start accelerating the processes involved in genomics and metagenomics. Some high-level examples or many more as well. And also a dream that we had in our research group, even like far before I joined, has been to have very lightweight embedded devices that can perform comprehensive genomics and metagenomics in real time. You know, as you might have heard or not, but there are sequencers called Minion that are as small as a USB device or like thumb drive developed, because there is huge need for being able to perform sequencing and analysis in remote environments because of many medical and environmental implications, or as even like in fields such as agriculture, to basically leverage these applications. So there's been huge efforts to enable sequencing in very light-based manner. So our goal and dream has been to also enable having lightweight computation as well, so that we can answer many types of questions in real time. I will talk now about some very high-level overview articles that you can look if you're interested to familiarize yourself from the efforts that have been done in this direction. So for example, so I have some here overview readings. Maybe also you can, of course, find the slides online. I highly recommend that we keep also updating these types of lectures. So if you're interested, please look at them. It gives a very good overview of some things that have been done to basically address some of the challenges. Let me also give you some, again, high-level examples, but by looking at some example papers or projects in this direction before going to the deeper works. Or like deeper analysis of some works. So one of the examples is GenAsm framework, a high-performance, low-power approximate string matching accelerator framework for genome sequence analysis. If you remember in one of the earlier slides, I was saying that to find the similarities and differences between the reads and the reference genome, it is needed to perform approximate string matching. and it's a very computationally expensive and challenging task to do. So this goal here in this paper has been to accelerate approximate string matching by designing a fast and flexible framework, which can accelerate multiple steps of genome sequence analysis that rely on this ASM. And it's the first acceleration framework first ASM acceleration framework for genome sequence analysis, and it's based on BiTAP algorithm. I will not go through the BiTAP algorithm, but the interesting thing I want to talk about it here is that it's an algorithm that in our conventional systems actually might not be a lot more effective than some other alignment and ASM algorithms. But if you consider the opportunity to design specialized hardware accelerators for them with very large, let's say, bit vectors here, you would actually benefit from this algorithm much more than some others that perform better on the CPUs. And so other things that are presented in this paper are highly parallel BiTAP with long read support and some novel algorithmic optimizations for traceback. I don't go through the details of what a traceback is here, but the important thing here is that this co-design is enabled by scalable and memory efficient algorithms with low power and area efficient hardware accelerators, meaning sometimes if you want to design some hardware units, you might need to, your trade-off space or your selection of algorithms or the way you optimize algorithms might be very different. So that's why I wanted to bring this example very quickly and also to show you what it gets enabled. Minimap2, for example, is one of the commonly used software algorithms in the field of genomics. And you can see that how this GenASM framework can provide more than 2 orders of magnitude of speed up compared to it. So the benefits that come with this specialization are huge with hardware units that are actually very low cost. There are also many more results in the paper. I will not go through them because I just wanted to give you a taste into what gets enabled by algorithm architecture co-design. You can see Damla's more detailed lecture here on YouTube, was the lead author in this work. We also have some follow-up work called Scrooge, which tries to, so it's a fast and memory frugal genome sequence aligner on CPUs, GPUs, and ASICs. So its goal is to build practical and efficient implementation of the GenASM algorithm for multiple computing platforms and compete with the state-of-the-art pairwise sequence aligners of that time. And it overall takes GenASM and tries to make it more memory frugal and address some of its inefficiencies. And also it provides some open source implementations for CPUs and GPUs. So if you already want to play with some of, let's say, off-the-shelf hardware, such as GPUs, and see what are the impacts of these optimizations and look at the code in more detail, you can already do that. I highly recommend, and it's all open source available on GitHub. And of course, you can also see more details about the talk and learn about the work and read the paper. This is another high-level example I want to give you is SegRAM is a universal hardware accelerator for genome sequence to graph and sequence to sequence mapping. So we have talked about read mapping in my earlier slide, right? And its importance of it in many genome sequence analysis applications. So we would have a linear reference genome. So this is a tiny difference, as you can see, but I'm just showing it for an example. So if you have different reads, then we can find the similarities and differences between this read and the reference genome. But in reality, having a linear reference genome is too simplistic because, so if you remember, again, in my earlier example, I was saying that imagine we have this healthy human genome and we want to map some reads from a patient to it to detect what's the source of some of the diseases or to find, let's say, genetic variations that can cause problems. But then in reality, we might have genetic differences based on our race or based on our geographic location, etc. And let's say a person from Europe or Africa or let's say Asia, they might have some differences that are not, let's say, related to a specific disease. So having a linear reference genome simplifies the problem just too much. And what many people have been looking into has been some graph-based reference genomes to enable sequence-to-graph aligners, sequence-to-graph analysis for a more accurate and fair type of analysis. And the interesting thing, again, the reason I put this example here is to highlight, like if you compare these two papers, SEGRAM, which is this one that targets this problem, and the previous ones that I talked about that focused on sequence-to-sequence alignment, you can see that, okay, there are some quality improvements in analysis, but analysis becomes more difficult, and the challenges that arise computationally in nature are different. So that, again, kind of highlights the need to look at algorithm architecture together, because based on some fundamental changes from the algorithm side, the problems that we need to address in hardware side can be different. Again, there are some summary of results that got enabled by SEGRAM, this work that works on sequence-to-graph mapping. It's actually a universal framework, so it also supports sequence-to-sequence mapping. But you can see, again, very large benefits provided by that. And more details of it, you can, again, go through YouTube. So this was some high-level overview of basically what this general domain is, if you didn't know about it, and why it is important for us to look into it, why we need to look into acceleration of it, and some examples of, let's say, benefits, like fundamental benefits of hardware-software co-design for it. So for the rest of this lecture, I want to dive a bit deeper with some of the examples. I am going to pick examples of storage-centric system design for genomics and metagenomics due to two reasons. One of the reasons is this. So we have looked at this system diagram, right, before in the previous slide. So we have a lot of heuristics, accelerators, and filters that try to alleviate computation overhead. But then the data movement overhead from storage system to all of these units still exists, meaning like we still need to move humongous amount of data all over the system to analyze. So that is one of the reasons that I want to go to my more detailed examples, the example of storage-centric system design. And the other example is that that's what the topic of my PhD dissertation will be. So I have worked on this direction a lot, and I would like to, by presenting each of these works, highlight some key aspects of algorithm architecture co-design. And I hope that these key aspects, you can take it with you beyond the context of necessarily these works. I will talk about an example for genomics. The system that we designed is called GenStore. And an example for metagenomics, and the system design there is Megis. Also, we've been focusing as a group on some emerging areas in genomics and metagenomics. So I will talk about GRAINS that works on genome graph analysis, and Mars on raw genomic signal analysis. And in the end, I will talk about efficient analysis on compressed genomic data, because that's also a very important thing to take care of. Okay. So again, as I said, I hope that we can have some takeaways from these beyond the context of these exact works. With GenStore, I will go through the details of it, but the key message I would like you to take away is the different trade-offs that we need to make when designing algorithms or like optimizing algorithms for a new different execution paradigm or new different hardware setup. So there would be some changes that actually might not be efficient in some baseline scenarios, but given underlying drastically different underlying hardware, those changes might be more efficient. So let's take a look at this work in a little bit more detail. Okay, I think. Okay, good. So read mapping, we discussed what it is. Let me talk about some high-level steps involved in it. Anyway, again, like mappers, different mappers might do things differently, but in a fundamental manner, they have these key steps. So we have this reference genome that we want to map the read to it, and the search space is quite large. So for a human genome, it can be more than 3 billion characters. Oh, by the way, in these points, if you have any questions, just feel free to interrupt me. You don't need to wait. Okay. So commonly, because search space is very large, we have this index generated offline with these k-mers, subsequences of length k, extracted from the reference genome with their locations. So we have this offline index generated. The first step is called seeding to determine some potential matching locations or seeds in this reference genome. So to do that, we extract these k-mers out of the read. So for pictorially, I'm drawing a read that is quite small, but the reads typically are larger. And therefore, that is why we need to extract these smaller subsequences of length k to be able to make it searchable. Otherwise it will be very difficult to search. Anyway, so we look up these k-mers in the index, and based on their positions, we mark some places as seeds. So wherever this read maps, it will be one of these things. Of course, then we need to go and do approximate string matching in these positions to find the exact similarities or differences, right? But again, because ASM or approximate string matching is super complex and costly, we don't want to go through each of them to do that. So we have something called seed filtering or chaining. And it prunes some of the seeds in the reference genome based on some higher level heuristics. So you can see some seeds are very far away from each other, right? So why would something map there anyway? So based on some high level approximations of that alignment process, one can easily get rid of some really, let's say, unlikely locations. So whatever moves through, we go to finally this expensive alignment step to find the exact similarities and differences using the approximate string matching operations. So that's it. I hope that you've understood the key steps. If you haven't, you can ask. Okay, I see some nodding. Good. So now let's go to some high-level motivation. I already talked about the motivation behind many of these works, but basically the case study that we did in real-world genomic read sets on various read mapping systems and various state-of-the-art SSD configurations, we realized that designing an in-storage filter, basically a filter that can already prune the reads that do not require expensive approximate string matching operations outside inside the SSD can help us reduce the computation overhead, because we know that, okay, anyways, I will talk what type of reads those things will be, but anyway, we will not need to bring the whole reads to the computation units anyway. And we also reduce data movement overhead. So meaning we reduce the computation burden the whole rest of the system and also reduce data movement overhead. And if we design the exact same filter to get rid of the reads that do not require complex ASM and put it outside the SSD, that provides lower benefits because it does not reduce the data movement overhead from the storage system. And also it needs to compete with read mapping for systems resources, such as main memory bandwidth and computational resources. And a hardware accelerator that is used for addressing computational bottlenecks in read mapping process makes I/O even a more visible or prominent bottleneck on the end-to-end performance naturally. So our goal, therefore, in this work has been to design an in-storage filter for genome sequence analysis in a cost-effective manner. And the design objectives would be to improve performance, so provide high in-storage filtering performance to overlap the filtering with the read mapping of unfiltered data, and have high applicability to support reads with different properties and different degrees of genetic variation in the compared genomes, and to be low cost. I will talk about these points in more detail. So as I mentioned, the system that we designed is called GenStores, that filters reads that do not require expensive alignment operation inside the storage system, so that we can only send the reads that get filtered not filtered to the rest of the system. And the challenges is that there are many different behaviors. Like what can you really filter? I will talk about it. Like there are different things that we can filter, and then therefore it is challenging to be able to support all of them. But given the fact that we have limited hardware resources in the SSD, so in a normal computing system, you might have a lot of main memory capacity and relatively much higher bandwidth to that main memory compared to the hardware units that we have available in our modern SSDs, which is the example storage system we are going to look into. So what do I mean by filtering opportunities? Let me talk about what the opportunities are, and then we can see how each of them would be designed. So sequencing machines provide or like produce one of two kinds of reads. One are short reads that are highly accurate and shorter, and long reads, which are relatively can be less accurate and are also longer. And reads that do not require the expensive alignment step, therefore, will be these things. One are exactly matching reads, because we do not need to do approximate string matching if something exactly matches, right? With a simple XOR, you can already find what matches. You don't need to do a whole dynamic programming for approximate string matching. So there is with low sequencing error rates, the short is with low sequencing error rates and low genetic variation. Prime example of species with low genetic variation would be us. Basically, us humans have very low genetic variation on average. A human read set has around 80%, a short read set has around 80% exact matches to human reference genome. So that's already a very important, let's say, class of genomic data. And the other one would be non-matching reads, where in the second step that we talked about, we filter seeds. In that step, if no seed remains, then there is nowhere to align anyway, so you can skip alignment. So either like high sequencing array, so that is one thing, but the important one is that the high genetic variation between the reads and the reference genome. Some examples of it would be if you want to do contamination filter, or the other example would be some species with very high genetic variation, such as some virus samples, then would benefit from this as well. Okay, so our system GenStore then has these two parts, GenStore EM and GenStore NM, for exactly matching reads and non-matching reads. So I'm going to talk about, now that we know basically what the problem is and what we want to do, I'm going to talk a little bit more about some changes in data structure and algorithm that need to completely be rethought of to enable doing some stuff in the storage system. So let's go. So efficient storage filter for reads with at least, so GenStore EM is an efficient storage filter for reads with at least one exact match in the reference genome. And it uses very simple operations without requiring alignment. The challenge is that we have a lot of random accesses per read to the reference genome and the index. So you have seen that we extract these k-mers and we need to look up the index and the reference genome, right? And handling that inside the storage system is difficult because of expensive random access to the flash chips, the contentions that can be caused, and also relatively we have less bandwidth compared to what we would have in main memory of, let's say, a larger scale system. And we have very limited DRAM capacity inside the SSD. It's typically used to hold some metadata. So for, let's say, 4 terabyte SSD, you would have 4 gigabyte of DRAM. So it's quite small. So, okay, we have very limited hardware environment to work with, but we want to find which read matches exactly to the reference genome. Okay, so that's a task at hand, and that's the underlying hardware. So let's see what we did here. So the first thing is that read-sized k-mers to reduce the number of accesses per each read. So we previously had the reads and its k-mers extracted from it. So let's say if you consider a short read of size 100 characters, and k-mers typically would be, I don't know, size 15 or 19, something in those ranges. So we extract these things and look up the index and then the reference genome. So we say that, okay, let's have this, let's call the whole thing a k-mer, you know? Like in that case would be if a read is size 100, then it would be 100 mer, right? So then we would have only one index lookup per read. And given that we know that per each read we have one access, then we can also already, when we are generating the read set and all, sort them in an offline manner to avoid random accesses to the index. Because the index is offline and sorted anyway, and then the read set, when being generated, it can be also sorted offline. So we can just sequentially scan through two sorted data structures to find their similarities, right? To see what matches to what. So at the end of the day, just a simple comparator and sequential scan. That doesn't come for free. I will ask you later what you think the overheads might be. But let me just show you in high level what I mean. So this is our read. Again, for simplicity, I'm showing just 10 characters, but in reality, it can be 100 plus. So this is sorted read table with the reads and their IDs. So I'm here for simplicity also sorting them based on their alphabetic order. And this is the k-mer index. If you remember, when I was giving read mapping background, these k-mers now would be with size of the read, so they have similar length. So these k-mers are extracted, the ones observed basically in the refresh genome and the locations that we observed them, right? And then again, yeah, both of them sorted. So then finding an exact match on such a thing is trivial, right? A simple comparator here, happy. We go, matches, yes, goodbye, filter. Then let's go check the next read and next k-mer, okay. The read is bigger than the k-mer, so we might need to check the next k-mer. Okay, now this k-mer is smaller than, is already now bigger than the read. So, okay, the read didn't match anything. So it needs to go to the read mapper to do the full job, you know? And now we go analyze the next read. So we avoided random accesses and we had simple low cost logic. Everyone is happy. What do you think the problem is? It will be too good otherwise. No, you find, right? I have all the reads, right? So then let's say reads the size 100. And then I have 100 mers extracted from all 100 mers extracted from the reference genome. So I have them. So if something happens in the reference genome, I will see it. But I have extracted all the 100 mers. Oh, then it goes to here, right? So if I didn't find any match, I send it to the baseline read mapper outside the SSD. So functionally, it's fine. Tell me. No, because because both of them are sorted, you go like this, right? So it matches, then I go to the next one, and then you see the process. So basically, the table needs to be read only once, and the read set needs to also be read only once. Basically, happily and simply, we scan through it only once. 0 random accesses. Okay, so the point would be that the read-sized k-mer index takes a large amount of space. So 126 gig just for human index. Why is that? Because you have a lot more unique k-mers, right? So like each of them are much larger than normal k-mers. And also each of them, so you have more k-mers and each of them are larger. So 2 issues. So the table becomes really large. And that is why in some baseline systems, this might not make sense to do it outside the SSD if you have a low bandwidth SSD. So in a host system connected to a relatively low bandwidth SSD, you might not want to bring all these to do some exact match filter. Maybe, I mean, if your reference, if your in the paper show that if your SSD has high bandwidth, this also optimization will provide benefits outside SSD as well. But yeah, in some cases it's not beneficial. But anyway, so two problems we said. One is that many k-mers and each k-mer is large. So let's address at least one of them. So we replace this with the strong hash value of these k-mers. We use some cryptographic hash functions for that. And it helps us reduce the size by around 4 times. It is still larger than the index we would have in the baseline case with the smaller k-mers, because still we have many more k-mers anyway. But the highlight I wanted to make is that sometimes based on our execution paradigm, what we need to optimize for might be different, right? In the end, of course, like with this optimization, we address a large part of this problem, but still like the remaining part is a trade-off we accept because of the huge gains. Like therefore, like by filtering like large amounts of reads inside the SSD, then you don't need to rely on computation units to basically do this task for you, like to move the whole thing from storage system to main memory all the way and get rid of a lot of data. So yeah, the key message I want you to take away from GenStore EM is that be open to think in a new way regarding optimizations when you think of completely new underlying hardware structure. And okay, so how does this look like? So this is a very high-level overview of GenStore-enabled SSD. So we have NAND flash arrays, SSD controller, that tiny little DRAM we talked about. These two tables, sorted read table and sorted k-mer index, are in the NAND flash array. They are stored across different channels, dies and planes, so that we can leverage full bandwidth. And very small hardware unit is added to the SSD controller. We access the batches of data like this. Actually, in our next work, we show that we don't need to actually bring it to DRAM anyway, but yeah, back then, that's how we designed it. And it goes through the comparator, and whoever doesn't get filtered, it goes to the host system or any other system that is doing your read mapping, right? So these two steps are pipeline, meaning when we are doing filtering in GenStore EM, we can send unfiltered reads to the host system, right? So, and think about the SSD controller, it already has a lot of complex hardware units in it. So just a simple comparator, of course, with some changes into the firmware, can help us basically get rid of a lot of data. And later on in some of our subsequent works, we show that some general purpose in storage units that can anyway support some simple comparison operations can be adopted to support these types of operations. But for that to be happening, we actually need to co-design these algorithms and architecture together. Meaning like dropping the conventional algorithms to it, I hope that you're convinced, would not be working easily. So that was my overview of GenStore EM. The next step is GenStore NM. I will talk about it in a higher level of detail compared to EM. But I am wondering if, Konstantina, should I explain this and we take a break, or? Okay, I explain this and we take a break, okay. Okay, so GenStore NM for filtering non-matching reads. So do you remember this 3-step thing that I talked about, like high-level overview of read mappers, right? So let's look at the second step. We want to basically see if there are cases where we can get rid of all the seeds in a read, and then we can skip the third step anyway. We don't need to go through expensive alignment operations. The issue is that how to perform chaining inside the SSD, because if you have a long read, we need to do some sorts of dynamic programming operations on many seeds. So if the read is very large and we would have many seeds, And therefore, we would need a lot of hardware unit to handle this, like a lot of buffer spaces to keep all these seeds, and to handle dynamic programming in all of them. So we were thinking that can we actually lower the cost of this step as well? So it uses a lightweight chaining filter that selectively performs chaining only on reads with a small number of seeds. So let me show you the key observation and anyway, it sends the more complex chaining stuff to the host. So this is very interesting. We looked at many species, like many types of classes of data sets to work with, and we made this observation. So the x-axis are number of seeds per read. Of course, this x-axis would also continue, but I chopped it off at some point, so we can look at this part, the earlier part. And the y-axis is alignment probability. And the observation is very simple, if you look at it. So if you have many seeds per read, then you most likely would align anyway. So you need to go to host system or like to any other mapping accelerator or whoever is doing your mapping to do alignment anyway. So why would we need to do chaining for those things inside the SSD if it needs to go through the whole computation anyway? So we then choose to do this selective filtering only for reads with a small number of seeds. So, and that on its own actually helps us to filter many non-aligning reads without actually expensive hardware resources in the SSD. I'm not going to talk about the hardware design of it because I mean, a lot of it is trivial, how from this point on would look like. But the key message again, I would like you to take away from GenStore-NM's design, hopefully beyond the context of just this one single paper, is that this cross-disciplinary knowledge between the fields can actually help some important challenges and important problems in very simple ways. So given this observation and this basically very simple insight, we can get rid of many reads and filter many reads in some use cases by very low cost hardware support. And simple operations. As I mentioned, some of them are so simple that can even be adopted on a general purpose in storage processing systems. So let me show you some results in high level. So we have base. Whenever I say base, I'm referring to a state-of-the-art software or hardware read mapper. These are the names of them for you can see in the slide. And GS in the plots means this base integrated with GenStore. So GenStore augments all these mappers. So a GenStore enabled SSD is augmenting all these mappers to help them filter the reads, right? So we will see how much it helps us improve the end-to-end read mapping performance. Okay, so we also considered various types of SSDs with various amounts of bandwidth. And here is a short read set with 80% exact matching reads. We see for software and hardware mapper, the speed up that is provided in each of these cases, and the large energy reduction. But also here, let me show you for GenStore-NM. So this is a contamination filter use case. So for whatever read set that you generate, it's important to ensure that there is no human contamination. If your sample is like for specific bacteria or virus, you want to map it always to human reference to get rid of any contamination that from the lab operators might have got to your sample, right? So we can see huge, like large amount of speed up can be obtained with just very simple designs and optimizations that we talked about, right? With this large energy reduction. We also looked at the hardware overhead of these things through some synthesis of these hardware units. So we are assuming that we would have our custom hardware for this at here, like a very old technology node. But anyway, you see that it requires very small area and power consumption. And if we use some scaling factors, we can see that it takes a negligible ratio compared to like a Intel processor at 14 nanometer, and less than 10% of the 3 ARM processors in a SATA SSD controller. It's important to note that on SSD controllers, we also have other hardware units. So it's a rich environment anyway. So this relatively, compared to just those 3 cores, it takes very small overhead. There are many other results also in the paper. So if you're interested in this direction, I invite you to check. And that's the paper. We keep more expanded versions of these in archive. So if you're interested, you can take a look. And that brings us to the break, after which I will talk about our next work. 15 minutes? Yeah. So, yeah, 2:20. Okay, can someone please check if I'm audible in Zoom? Okay, great. Okay, so now let's discuss with the next part, which is going to be about Megis, our storage system for metagenomics. And the key message I would hope that you would take away after this presentation is how we can basically do this co-design across the system so that basically we leverage the strengths of different parts of the system to address some of the weaknesses. So it's about, let's say, a more broader co-design throughout the system. Now, let me remind you of this pipeline again. I've talked about it before in the background section, but I see some few new faces, just a very brief reminder. So we need to figure out what is in our sample. We need to prepare our input queries by extracting some smaller subsequences of length k called k-mers. So we have these k-mers that are easier to search. And then in the second step, we look them up into this large database containing information on many species and figure out what are the species that are present or absent in the sample. And then the third optional step would be abundance estimation. There are also other downstream tasks one might want to do. But this database can be very large. In some cases, it can be more than 100 terabytes even. So because that we need to deal with large amount of data, we perform some motivational analysis and case study of performance of metagenomic analysis tools with various state-of-the-art SSC configurations. Let's see what this plot is showing. Compared to so on the y-axis, we have normalized throughput. On the x-axis, we have two different databases. So compared to an idealized scenario with no I/O overhead, in a case with performance optimized and cost optimized SSDs, we are seeing that in both scenarios, there is a large I/O data movement overhead that causes throughput and performance degradation. And actually, similar to before, I/O becomes even a larger overhead in systems where other bottlenecks are alleviated. So this I/O overhead due to accessing large amount of low reuse data is hard to avoid. And this slide actually is not necessarily related to metagenomics, but in any type of low reuse data that can apply. So let's see why. So first is that, okay, you can do some sampling techniques to shrink the database sizes, but that can lead to accuracy loss, particularly in this case, because in many scenarios, actually, it can impact accuracy in levels that are unacceptable. You can imagine some clinical scenarios where accuracy loss is simply not tolerated. The other potential solution might seem to be like, one might say, okay, let's keep all data required by metagenomic analysis completely and always in main memory, right? But that is energy inefficient, costly, unscalable, and unsustainable. Why is that? So these database sizes that are already very large are increasing in size rapidly, like doubling every few months. So why is that, by the way? So we don't know a lot of species that are covering the surface of Earth. And after we get to know them, we need to add them to these databases. And the other point is the fundamental nature of many species evolving. Like we have, over the past few years, read how viruses evolve really quickly, or like bacteria. So as things evolve, or as we also discover newer things, completely newer things, then the size of databases by definition need to increase in size. And the other important factor is that different analysis might need to access different databases. So not like we cannot basically rely on keeping a single database at memory at all the times, because as I said, like based on the use cases and what we want to analyze, we might need to access different databases. So therefore, because of all these reasons, it's very inefficient and unscalable to go with this second potential solution. So motivated by these points, our goal is to improve metagenomic analysis performance by reducing large data movement overhead from the storage system in a cost-effective manner and with high accuracy. So what are the challenges? Of course, like we have learned about in storage processing in the previous talk as well, right? So no metagenomic analysis tool can run inside the storage due to SSD's limitations. For example, long latency of NAND flash chips. Because of these reasons, like limited DRAM capacity inside the SSD and limited bandwidth of that little DRAM anyway. So in this work, we try to address these problems, and we propose MEGIS, metagenomic in storage. So it's a first in storage system for end-to-end metagenomic analysis. And its idea is cooperative in storage processing for metagenomics, which is enabled by hardware-software co-design between the storage system and the host system. So we basically want to make these two help each other so that we can kind of address the limitations of each of them. But of course, there are a lot of challenges that arise for making these two together do the job. So these are the high-level steps of MEGIS. So step 1 would be data preparation, step 2 would be presence-absence identification, and step 3 would be abundance estimation. Now I'm going to give you a high-level overview, some hardware-software co-design that we had to do to address the challenges of making these two working together and to basically leverage the strengths of both. So I'm wondering if anyone could guess what would be the challenges of making these two work together? There might be several challenges. Some of them be addressed in this work and we mitigate its overhead, but any guesses? Just for me to remind you again, we don't need to incur IO data movement overhead if we do stuff inside the storage, but then we have very limited DRAM capacity and bandwidth, and we have this slow NAND flash chips inside the SSD, and we don't want to also do too much writes to it because of lifetime constraints. So it would be good if we can make both of them work together, but what would be the challenges of them working together? I heard someone saying communication. Yeah, okay, that's correct. Whoever said it, yeah. Okay. Okay, yeah, cool. Anyway, yeah, that's a good problem, correct problem. So the first step was task partitioning. To like first aspect of our code design is to make them each step run on the system that it benefits the most from it, and then the other one is to have some data computation flow coordination to reduce communication overhead between these and reduce the number of writes to the flash chips. It would be counterproductive if we divide the steps to where they work best, but then have a lot of communication between them, right? So it is very important that when we are designing such type of heterogeneous system, we ensure that we don't get like diminishing returns because of communication between different parts. I will talk about it more about this point in my slides, in my next slides. But anyway, another key aspect of our hardware software co-design is, again, storage-aware algorithms because of enabling efficient access patterns to the SSD. So you've heard about same type of things in the previous design that I talked about. Then also the fourth aspect is lightweight in storage accelerators to minimize SRAM and DRAM buffer spaces needed inside the SSD. And finally, the last one is data mapping scheme and flash translation layer that is specialized to the characteristics of metagenomics analysis, such that we can leverage SSD's full internal bandwidth. So we can actually handle how the data is laid out inside the SSD based on how we know it will be accessed in this class of applications, right? So that we can actually have more efficient access patterns and leverage the available bandwidth that we have. Okay, so I'm going to talk about some aspects of these by talking a little bit about these steps. So step 1, data preparation for input queries. It employs sorted data structure, again, like to avoid expensive random access to the SSD. So we extract the k-mers from the sample and sort the k-mers. The database is already sorted offline. So you might wonder why in GenStore we already had sorted read sets. Why can we not have sorted k-mers here? Because here we are extracting k-mers out of the reads. So we need to not only store the reads, but then in that case, we had to store the k-mers as well alongside the read, but that would be huge because as you see, k-mers have a lot of overlaps with each other, and the k-mer space is much larger than the reads themselves. So it would not be effective that for each single data set be stored sorted k-mers as well. Also, what k-mer size are we going to work with? Depending on application, that might be different. Okay, so Megis executes this step in the host system. Why is that? Because it benefits from larger DRAM and more powerful computation, because we need to extract a lot of k-mers, so there's a lot of writes involved. It's good to have this DRAM because otherwise we need to write to the NAND flash chips so they can hurt lifetime. And also, yeah, as I said, so it incurs fewer writes to the NAND flash chips than the case if we were to execute this step inside the SSD. And it also enables us to overlap step 1 with step 2. Step 2 is going to be in the host, in the storage system, as we will discuss later. So doing this step in the host and benefiting from these capabilities also enables us to overlap these steps to run in concurrent pipeline manner. But to actually be able to do that efficiently in the host system, we need to avoid significant overhead due to data transfer time between the steps, as we talked about, and also minimize performance and lifetime overheads even when host DRAM cannot hold all the query k-mers. So this is how it works, step 1 in our design. So host CPU, we have query k-mers, so divide the k-mers. So what we do is that we divide the k-mers into independent partitions based on their alphabetic range. So this green part is the Megis-enabled SSD. This part, I'm not expanding it because this is not executed in the SSD. This is in the host system, is where I'm expanding it, host CPU and host DRAM. So we extract the k-mers and then write them into alphabetically partitioned ranges. We have more partitions. For simplicity, I'm just now showing a few of them. So what does it help us is that we can overlap operations on different partitions. So for example, once we are, let's say, doing sorting on C, partition C, we can already move partition A to the SSD and do computation on it. So it hides some of that computation overhead, right? And the other benefit of it, I'm not showing in this slide, but let me briefly talk about it. Because we are working with these partitions, if the DRAM space is not large enough in the host, we can pin some of it to DRAM in the host and pin some of it in the SSD. So at least this way, we don't need to have frequent swaps for all k-mers all the time. So that's another benefit. So actually, this technique is very useful for leveraging the best of both worlds while hiding the communication between them and also not incurring writes to the SSD. Okay, step 2. This is quite simple. So we identify the common k-mers between the query k-mers and database k-mers. It's a little bit similar to GenStore EM in nature, if you think about it. The second part of it, though, is different, is to retrieve the IDs of the species. So we have this input query with some k-mers. We have this massive database with some other k-mers. We find out their intersections, and we want to see what does this intersection mean? Like, is it a specific virus, bacteria, or whatever, right? So the IDs of those k-mers can tell us what is actually in the sample. So Megis executes this step in the SSD because it accesses large data with low reuse and involves lightweight computation. And to execute it efficiently in the SSD, we need to leverage the internal bandwidth of the SSD effectively and not require expensive hardware resources. As you remember, we're working in a very tight environment. So let me show you here. So now I'm expanding on that SSD thing, right? So this is a Megis-enabled SSD. The yellow part is SSD controller. In the SSD channels, we have the database k-mers happily stored, and then we have the SSD's DRAM. If we go with what we did before to bring this database k-mers to the DRAM and then bring the query k-mers from the host system, because now we're working with both of these things, right? And also writing the results, what we end up doing is to pressure that limited bandwidth. So it cannot afford actually doing all that. So what we do is that we adopt a very simple approach to compute directly on the flash data streams. And actually, because the operations we need to do are super simple, we can reduce this buffer sizes significantly. So we can have just 2 simple registers per each SSD channel. So that one is to have the camera that we're working with to find the intersection of it with the database, with the queries. And then the other one is for the next camera coming from the controller. So it's a very simple double buffering thing going on with very small overhead. And then we write the result. Okay, so we have the common k-mers. We are happy. And we want to find the species IDs of the common k-mers by looking up a sketch database. So what we do here is that, so each type of species would have different sketch k-mers, like k-mers that are representative of that species, like a signature of it. So we can have then in our sketch database various k-mers and their IDs, right? But in reality, this takes a lot of space that for us to be able to store all these sketches and their IDs. So what people have been doing in the community is to come up with more space-efficient ways to store this data. But the issue is that a lot of these solutions are very slow for adoption inside the SSD due to long latency of NAND flash memory. Okay, so we have a problem here. And we again try to think of a solution that is fit to this new execution space inside the storage. We call our solution k-mer sketch streaming. I'm not going to go over the solution because it takes a little bit of time and detail to go through exactly how it works. But if you're interested, I really invite you to check our paper. But what it happens is that actually it's quite a lot smaller than, let's say, space inefficient or naive ways of storing the k-mers, while it's still larger than these state-of-the-art space-efficient solutions. But actually what it does is that it's much more suitable for our in-storage processing paradigm because, again, it enables us to figure out these k-mer sketches and find the IDs only with streaming accesses. It has a different design than what we have discussed before. So I invite you to check our paper for its data structure. But again, what I want to highlight again and again is this completely different trade-offs that one needs to make. And the benefits that come from basically knowing, having knowledge about intersection of both domains, right? So you can enable fundamentally more efficient solutions. Okay, so step 3, again, the design of it along with our FTL design are in the paper. I invite you to check it. And let me talk about some of the results. Yes, please. Future of SSD? You mean like, can we still use that SSD as a normal SSD? That's a very good question. Yes, because these augments the SSD with some minor changes to its FTL and to its hardware units, while preserving the baseline functionalities. So we would have some commands from the host system that sends to the SSD to say that, okay, now you're entering your acceleration mode, like metagenomic or genomic acceleration mode. But before that, it can work with as if it's a normal SSD as well. Yeah, sure. Okay, so I'm going to talk about some results, but I will, at the end, highlight a particular result which I really like myself to hopefully also propagate to you what are the fundamental benefits one can provide by these types of designs. Anyway, our methodology, so we have performance, energy, and power analysis based on hardware and software components. You can see the features and the tools used. So based on comparison points, we have performance optimized software, accuracy optimized software is using also a larger database compared to the first one. And PIM hardware accelerated tool, which uses processing in memory to further accelerate the performance optimized tool. Meaning it uses processing in memory to perform k-mer lookups. Okay, so, and then we use again different SSD configurations with different bandwidth parameters. Yeah, okay, so it seems like I only kept this result anyway, my favorite result. Anyway, this can help us see something. I'm going to evaluate these baselines in two different types of system. One is a cost-optimized system with lower bandwidth cost-optimized SSD and 64 gigabyte of DRAM. And the other one is a performance-optimized system with the performance-optimized SSD with higher bandwidth and 1 terabyte of DRAM. So it's a huge difference between these two systems. So in this plot, I'm going to show performance-optimized and accuracy-optimized baselines on the cost-optimized system and the performance-optimized system, comparing it to McGis on the cost-optimized system. Meaning basically, for baselines, I evaluate them in both the cheap and expensive system, while McGis runs on the more expensive one. So here, we're already seeing performance of these. And this is McGis. So we see that if we do apple-to-apple comparison, we would have a lot of benefit from McGis. But the interesting thing is that McGis outperforms the baselines even when running on a much less costly system. This is interesting because if you remember earlier, I was talking about the need for having portable analysis, right? So there are many remote areas, both for medical and also biological, let's say, environmental or agriculture reasons. There is a need to perform genome analysis, and that's why from the sequencing side, people have been designing portable sequencers. So as I mentioned, we have sequencers as small as a thumb drive to generate digital data for us to analyze from the samples. But then this analysis actually shows that on a significantly lower cost system, and even such system can be portable, right? Even a laptop can have that much DRAM. You could perform more efficient analysis than relying, let's say, on a server node with 1 terabyte DRAM, etc. So it's both higher performance and more energy efficient, which I haven't shown you, by the way. But yeah, the result also shows much higher energy efficiency. And also one thing to highlight is that McGis achieves the same accuracy as the accuracy optimized baseline. So yeah, it also improves therefore this cost efficiency, right? And we really hope that such type of designs in the long term, of course, like when there is new hardware and new, let's say, execution structure for its proposed, it takes time to adopt, but we think that this direction can help enabling wider adoption of these really data-intensive tasks. Okay, and there are more also in the paper. And now I can go to our emerging areas that we have been focusing on as a group. I will very briefly talk about grains. It's an ongoing work we have on genome graph analysis. And then after that, I will talk about Mars. So we talked about graphs early on in the lecture as well, but now this is, let me remind you a little bit. So if you have different input sequences, a graph representation of it can provide really powerful and efficient representation of underlying genomic data because it has greater expressive power and it is more compact representation, which becomes very important in massive population scale databases. So if we want to do some analysis across the whole population and want to encode genomic data of every single person, then such type of graph-based representations have been very fundamental in enabling this type of analysis. I don't go through the details of our work, which we call grains, so it's for graphs and storage. But what I wanted to highlight is that what it achieves very quickly. So it's a more, so we enable it by more storage-friendly analysis, by specialized execution flow and scheduling, and further improve performance, energy efficiency, and cost effectiveness via in storage and in flash processing. So we, the works I've talked about had some hardware units on the SSD controller. Here, we also devised some little hardware units on the NAND flash dies. And it comes with its own set of opportunities and its own set of challenges. I'm not going to go through it now, but again, like I wanted to show that the large benefit it would provide. And again, similar to what we showed for Megis, we showed that it can provide these benefits without even relying to bring these massive graphs to a large DRAM in the host system. So now for Mars, I want to talk about it in a little bit more detail. Mars is looking into a very new direction in genomics, very interesting. It was led by Melina and Konstantinos here. I had the opportunity to also be part of this paper. So I will talk about our work together. So we discussed some high-level takeaways that I would like you to have in mind after discussing each of these. So for Mars, what I would like to emphasize is to kind of like the very new way that it has to look into the underlying hardware. So it takes an SSD and not only does computation in it, but it leverages the hardware units that are in the SSD for computation as well. Because in the lecture, you might have heard about computation in processing in memory. So it uses some memory, the DRAM that we talked about, in a less passive way, in a more, let's say, computation-enabled way. And in reality, because the storage system is a very rich environment, right? It has this NAND flash memory, it has DRAM in it and some computation units. So it tries to take advantage of the richness of that substrate in a completely new way. And I hope that we can look at it in high level through these slides. But let me first tell you what is raw signals, because this is also working on a new domain. So this is a sequencer. That's an example of the little thumb device that I talked about. Anyway, so these are different pores, or like nanopores, then the DNA strand goes through them. And then the sequencer outputs some signals like this. So the raw signals will be these ionic current measurements generated at certain throughput. And raw signal genome analysis, what it is? So let me first tell you how the normal analysis was. So we have this raw signal, have this process called base calling that generates this ACGT characters that we use to represent different nucleotides. And then you have seen the rest, right? How we do read mapping. In some cases, the sequencing and base calling or read mapping, you might want to analyze it multiple times or with different parameters or different references. That's a different story. But in some cases, you might want to do the runtime analysis. But also overall, there would be benefits if you could do that, right? It would be nice. So it contains some richer information than the bases in some cases. And also, it enables efficient analysis with better scalability and portability. So if you want to do some real-time analysis, then we don't need to rely on that, let's say, expensive process to translate things. We can just do this. So the raw signal genome analysis is this novel approach that operates directly on these raw signals, right? Some high-level overview of it, how it looks like. Let's think about it in a very high-level way for now. Fundamentally, it's very similar to what we talked in the past. So we have some indexing offline, but now we need to index the raw signals. So we have a reference genome of some humans, or I don't know, whatever other species that we want to work with. So it needs to be translated into these chunks of signals and hashed. By the way, how to hash these things is an interesting direction that we have done in our group. I will have the link of these papers later on in my slides. Because it's very interesting to think about it, how to hash these signals accurately. So we have a series of works on what we call raw hash to hash the raw signals. But anyway, that already exists for now in this figure. So this table is generated in advance, and then now mapping, which would be online process, is very similar to what we talked before. So we would have some little sequences. Here we have the signals to hash them, query some index, and then go and do some chaining. Chaining would be that lightweight second step that I talked about. And then after that, you can optionally move to alignment, which is a direction also we have worked in the group to do raw align, basically, to align the raw signals. But anyway, here would be basically the overall flow of it. Again, so this is the part that we want to focus on. So we have these chunks, like these events that we detect, okay, these are the chunks we need to work with. And the second step, seeding, and then chaining. And the third, alignment step, which in this work, we're not going to support alignment, but we will be covering these 3 steps. Okay. Some motivation is a little bit like in common with the past that, okay, sequencing throughput is very large, but then relying on conventional systems would be bottlenecking the whole thing. So RSJ, or like raw signal genome analysis, so when I say RSJ, then I'm referring to raw signal genome analysis, struggles to keep up with increasing real-time requirements, and we cannot keep pace of the rate at which this data is being generated. And there is critical need for acceleration. Actually, a few weeks ago, I was attending a clinical metagenomic conference in Geneva. I was talking to a medical doctor who works in ICUs in NHS. And he was saying that they have the need to perform raw signal genome analysis because it can help with reducing sequencing costs and improving the overall throughput for some of these ICU use cases that they have, ICU patients, because the faster you are, the better. So just imagine you have a patient that the normal blood test or like medical imaging have not revealed what is wrong, and you cannot have a treatment. So things like metagenomic and genomic can lead to much more accurate understanding. And therefore, it is very important to facilitate this real-time analysis. But then they were complaining about the fact that it's very difficult to do efficient real-time analysis because by the time you would be able to analyze the sequence chunks is already too late to show like a good benefit for the raw signal analysis. So yeah, it was basically interesting for me to hear the problem from, like, in the wild, in the field. Okay, and theoretically, we don't want to also use this for ICUs or like only for critical cases. We want to enable adoption of these things in our day-to-day life, right? So you go to medical doctor, how easily you can do a blood test. You want to already be able to do this type of tests at a low cost. Sequencing is already becoming cheaper and cheaper, so it's kind of important to ensure that computation becomes cheap as well. Anyway, okay, there's a critical need for acceleration, and the goal is to identify the key performance bottlenecks in RSJ pipeline to target the most time and energy intensive steps for hardware acceleration. Here is some breakdown. You can see, so for I/O, event detection, seeding, and chaining. So chaining and seeding consistently dominate runtime, and event detection and I/O overhead are also non-negligible in some cases. But then the issue, or not the issue, like another key message I want to tell you is that let's look at this data movement overhead when using some of the accelerators that we have already for seeding and chaining. Because as you know, seeding and chaining are some common steps, right, in genomics. So if we were to use accelerators based on how much they would reduce your accuracy, we would see that the IO percentage would also increase. So it's also a key message you have probably heard several times in this presentation today. So as compute overhead gets alleviated, IO becomes the dominant contributor. That this work tries to basically look at both cases. So IO data movement and the computation. So design a high performance and energy efficient system for RSGA by addressing both of these. And that brings us to Mars. Okay, so I hope that by now I've motivated you why this is an important field and why we need to accelerate it. We will now go through the design of it, but let me remind you, the key message here is to learn this unique way to look at the storage system, because one way is to keep looking at it as like a passive device that is just storing data and goodbye. And the other way to look at it is to kind of like be conscious of the very rich environment that it's already providing us and use it for accelerating these important tasks. And I hope that basically this would motivate us overall to look at the storage system as a more active and smarter device. So it's Mars is the first storage system for RSGA, and its key idea is a storage processing system that addresses IO data movement overhead and exploits SSD's parallelization capabilities to accelerate computationally expensive steps. And it's a co-design between targeted software modifications and dedicated hardware components within the storage system. So the target software modifications are to optimize RSGA for ISP, or like in storage processing, tailored to SSD's parallelization capability and architecture. So again, what we need to do, what we were also doing before, but in new ways based on features of this algorithm. And it's also enables processing in memory within the SSD to accelerate the RSGA steps. So it leverages from processing using the SSD's internal DRAM and also near that SSD's internal DRAM. Yeah, so the unique part basically would be the second, of course, the software optimizations are also new, but the interesting thing is the second part, because we are using the existing structures in the SSD and envisioning some lightweight changes for it to enable some great benefits. So how does Mars's workflow look like? So there are some software modifications to reduce computation overhead and intermediate storage requirements. So some filtering techniques to discard candidates that likely result to invalid mapping and include some frequency and voting-based filtering. And arithmetic conversions, just to convert floating point to fixed point, and reduce memory footprint of computational and computational costs. But the challenge, of course, has been to preserve accuracy in the presence of this raw signal, which is already noisy. And it requires careful placement of both of these techniques in the pipeline and having fine tuning of these parameters so that we don't actually lose accuracy, because as you know, accuracy is an important thing in this domain. And the paper conducts, so in the paper, we have analysis to show the software modifications do not have adverse impact on accuracy. So the ground truth is a system that uses base calling and read mapping. So base calling was a process that translates these raw signals to ACGT, right? So we are going to use that as our ground truth. And then the data sets are with different species, as you see, like diverse species. Each of them have different reference genome sizes. And the comparison point would be RowHash2, which is a state-of-the-art raw signal genome analysis algorithm. And Mars on CPU with just this first software optimization, and Mars on CPU with both of these optimizations. And a key observation is that if you look at this table, like I can just summarize the observations for you. It improves the recall and F1 score over RowHash2. So there are these filtering techniques are effective in eliminating ambiguous and redundant candidate matches and allow pipeline to focus on more informative regions that are more likely to represent a correct match. And the second part that we can focus on is that adding both optimizations here. So using fixed point arithmetic in the smart CPU only leads to minimal accuracy loss. So we apply earlier normalization and quantization to ensure that these fixed point arithmetic has no significant impact on the result. So it's this part is like pure software, and as you can see, it can be adopted on our current systems as well to provide some benefits. But as another benefit of it is that it can also reduce the workload of some of these in storage units. So now let's look at the architecture and system. We have two modes. So conventional mode, the SSD can perform as standard operations, that is, as it is a normal SSD. And then acceleration mode, which is, as I mentioned, as a response to the question, like this also applies to the previous works as well. Like we have these two modes, right? So this acceleration mode SSD executes as a pipeline to accelerate RSGA. So let's take a look at this. So we have the SSD controller and then a flash memory and the SSD's internal DRAM. And we have here flash controller and the units that we will talk about more. So let me basically go through these things one by one. So Mars control unit is just like a simple final state machine that orchestrates pipeline execution and control and data flow. And then we have the sorter and merger unit. So sorter performs parallel sorting of sequences, and the merger combines the sorted subsequences into larger sorted ones. And it is executed near the DRAM. So one sorter or merger, we assume that for one flash controller, we have one pair of sorter merger, and it's located near the DRAM. So less data movement basically between where the data is and these sorter and merger units. Then we have a simple arithmetic unit that performs arithmetic and logic operations required for RSGA. And again, it's located near the DRAM based on some previous near DRAM architecture, Fulcrum, to enable low latency and also high parallelism that can be enabled in this case. So it's one per each two subarrays of DRAM. So as you might have seen in the previous lectures, performing computation close to DRAM can provide large benefits. And as you have seen in this lecture, performing computation inside the SSD can provide benefits. But then here we are exploring, okay, why not all of them combined, right? Because we already have this rich substrate inside the SSD. And that comes with the freedom of basically being able to look at this SSD not as like a passive device, as I mentioned, and to be aware of its richness already. And finally, we have this, like next point is we have this query unit that performs efficient in-memory hash table lookups for seeding, and it's a processing using memory based on Pluto architecture. You can check these works separately if you want. Leveraging DRAM row activation for massively parallel hash table lookup operations. So yeah, this work, as you can see, has a completely different flavor than the previous works because it tries to more aggressively re-architect the system and think of it as a new way, in a new way. So we looked at different capabilities and different tasks, and then so we need to map this pipeline to this architecture. So we break down RSGA steps into fine-grained tasks of arithmetic, querying, and sorting operations, and implement Mars as a pipeline of these tasks, and each of them is mapped to a dedicated compute unit. And its control unit encodes a pipeline and enables this execution flow with a finite state machine. And basically it signals how each of these tasks basically need to move through this substrate. Okay, yeah, basically now then the execution flow would be very simple, right? So we have the raw signals being read and hash values are generated, queried, and then we have this batch of sequences to be sent to the sorter, then merged, and then we can do the chaining operation on it using a simple, lightweight dynamic programming. And a lot more details of it are also in the paper. Again, then the key message was how we could, let's say, leverage various types of execution paradigms inside the storage system. And the cool thing here is that if we have some flash-enabled, like not flash-enabled, like computation units integrated with this flash memory, or like, let's say, in a lightweight system with these SSDs, it can then very quickly plug the sequencer to it and perform analysis without relying on expensive computation units, like porting all this data to the cloud and relying on many server nodes to be keeping up just with these computations. So it's basically some steps making us go closer to the dream that I mentioned earlier. And more details about the mechanism is in the paper. So if you're curious to see how such system that leverages all these units in the SSD can work in detail, please check the paper, and you can, of course, contact us, the authors, as well. Let's look at some evaluations. So we compare Mars against 4 systems for performance and energy analysis. So the first one, BC, is the one that uses base calling, basically to generate ACGTs first and then doing read mapping. Each of them are using conventional GPU-based and CPU-based state-of-the-art tools. Then we have GenPeep, is a non-volatile memory-based hardware accelerator pipeline for base calling and read mapping. So it's using acceleration primitives outside the SSD in this case. So we can, by looking at that, see what's the benefit of doing things in SSD anyway. Oopsie. Okay, so we have RowHash2, the state-of-the-art CPU baseline for RSGA and Mars CPU, all of these things, optimizations only in software, and the full-fledged Mars, basically, that uses also these hardware units in the SSD as well. I don't go through the methodologies. Mainly, some parts of it are similar to what we did before. You can also check it in more detail later. So let me just highlight some results. Okay. So you can see all these different baselines here, and then this orange line would be RowHash2. So Mars performs all other baselines across all datasets, and it outperforms GenPeep that adopts PIM outside the SSD because it is addressing the IO data movement overhead that cannot be addressed no matter how much you would improve computation overhead outside the SSD. And Mars CPU on its own also provides some good benefits over RowHash2, which is also a software baseline because of the software modifications that were effective in reducing computational loads. Again, similarly, we compare these baselines, but this time for energy, and we show that it reduces energy consumption over all other baselines across all datasets. And the improvements stem from reduced execution time and also reduced data movement overhead. And in conclusion for this work, Mars is the first in-storage processing system for end-to-end raw signal genome analysis, and it enables it's enabled through software and hardware improvements. It provides very high accuracy and large performance and energy benefits. As you can see, several orders of magnitude in some cases. And again, as I mentioned, there's a lot more in the paper. I invite you to check. Some cool things. I really like this paper, so I really recommend to you to check. This is the paper Raw Hash, enabling fast and accurate real-time analysis of raw nanopore signals for large genomes. And the interesting thing is that this paper, for the first time, came up with an effective way to hash these raw signals. Why, and why was it challenging, and why? How did it get addressed? I really invite you to check. You will enjoy it. And this RowHash2, which are some improvements built on top of RowHash to make it even faster and more accurate. And RowAlign, which is basically enables alignment on raw signals. It's also very interesting because the conventional algorithms used for aligning these ACGT characters would not work when you want to align raw signals. And this is also RowHammer. It's a newer work which is enables overlapping and assembling raw nanopore signals using hash-based seeding mechanism. So you can enable, you can imagine how much it can get enabled, but just like a key idea to be able to hash the raw signals. Yeah, so that was it. I'm wondering if we should take a break or not, because I have very few slides left. Maybe we can just go through the whole thing, right? I hope you guys bear with me. Okay. Now this is the last work that I'm going to talk about, and it's like very high level again. So I will not go through the detail of it as much as I did with prior works, but I want to talk about the new problem here. So genomic data is growing at a very fast pace because we have these high throughput sequencers with very low cost, meaning the cost has dropped significantly, their throughput has increased a lot, and the rate at which we've been generating the genomic data has been increasing exponentially in public and private databases. So that is why there's been a lot of effort to accelerate genome sequence analysis. This is what I've been talking about over almost the past 2 hours, right? But also at the same time, there's been a lot of effort to store this genome sequence data in a highly compressed way. So the overhead that we show here is a data preparation bottleneck where genomic sequence data is stored in compressed form and needs to be decompressed and formatted first before an accelerator can operate on it. Let me talk about this problem with a simple pipeline. So this is the baseline. So over different batches of data, we prepare the data to feed it to some analysis pipeline. And then same for the next batch, etc. Okay, and then we would have accelerated analysis. Sorry, let me go back. So now here we would have the accelerated analysis happening, where the purple part has been significantly shortened. Many thanks to all these optimizations we've talked about, okay? But then we're bottlenecked by the yellow part. So just to prepare the data to feed it to these analysis pipelines. And now then we have accelerated analysis with idealized preparation as a hypothetical case. So if we were to accelerate this or yellow part, what would have happened? So we see that there's a large potential benefit for genome analysis acceleration. Actually, this plot is not at all to scale because you wouldn't be able to see the whole thing. You can see some example throughputs up there, but yeah, the benefit would be huge, but a large part of it actually gets lost just because we need to prepare the data to feed it to these accelerators, okay? Okay. So our design stage is an algorithm architecture co-design for highly compressed storage and high performance access for large scale genome sequence data. So what it does is that we carefully exploit the properties of genomic data in synergistically co-designing algorithm and hardware such that we have high compression ratios comparable to state-of-the-art genomic specific compressors. So genomic specific compressors are different from general purpose compressors because general purpose compressors are very ineffective in compressing genomic data because they fail to catch the long-range similarities in DNA data. So Sage's design is having compression ratios similar to these genomic specific compressors that are a lot more effective for genomic data, and it enables fast decompression only using lightweight hardware and efficient streaming accesses. So it can be integrated with genomic systems, even in resource-constrained environment, meaning we have put a lot of effort into designing accelerators for genome analysis, but then can we prepare the data in a fast and efficient way for them in a lightweight? So that's what we try to enable. And I wonder if you have any examples in mind for resource-constrained environments. Like, why would that matter? You might have heard about some of it today. Yeah, correct, definitely. meaning portable systems for portable analysis. With the other case, basically near data processing systems, for example, in storage or in memory. So I talked about some in storage systems, right? And we already saw a lot of hardware limitations there. And there are also some in-memory, very nice designs for in-memory acceleration of genomic data that provide large benefits. So it would be very challenging if you want to rely on expensive hardware just to prepare data for these lightweight accelerators in these resource-constrained environments. So Sage improves the average end-to-end performance and energy efficiency of genome sequence analysis accelerators a lot. So it can be combined with them to help us actually unleash the benefits that they could actually provide by basically removing data preparation outside the critical path. And there are more in the paper. We will be soon also updating this link with some additions. So overall, I hope that I could have convinced you that storage-centric systems for this class of applications can provide large benefits in performance, energy efficiency, cost effectiveness. And while I didn't talk about it also for robustness and security and privacy, and make accurate metagenomic analysis more accessible for its wider adoption, so that it's not just something that some fancy institutions for very critical cases would use. So it will be something that is affordable for all of us to use in our day-to-day life. And as I said, also the reason I talked about these things was to give you a little bit deeper sense, or like a more accurate taste of this hardware software co-design for these domains, and what knowing basically both domains, meaning computer architecture and genomics and metagenomics, can enable. Okay, so now I'm going to do a little bit of advertising. Bear with me. So we have this initiative that me, Onur, and Konstantina here have been trying to start, and we hope to keep expanding it a lot more. It's called Architecture for Health. And we had already a workshop of it in micro conference in a big conference in the field a few months ago in Seoul. And the goal is to basically explore key computational challenges in health-related applications and the vital role of computer architecture in overcoming them. You can find out the agenda and also the recording of the meeting and the whole workshop online. We covered many different domains like genomics, proteomics, variable systems, brain-computer interfaces, and agent-based simulations. Basically, the opportunities is that we are now presented with this wealth of data because of recent biotechnological advances to have high throughput, low cost, and accurate biological data generation. We talked about genome sequencing today, but there's also continuous variable sensing, medical imaging, etc. And this wealth of data enables unique opportunities for advancing healthcare, right? Because of precision medicine, bedside personalized care, discovering early warning signs of communicable diseases. Basically, we don't want, like if we next time see a random virus somewhere, we want to be able to detect it soon enough so it doesn't travel to the whole planet, you know, hopefully. And it is important, right? Like these things are something that preemptively we should be working towards and collecting data and be able to keep up with the analysis of it. Continuous physiological tracking, enhanced diagnostic capabilities, etc. But again, the challenges are very similar to what we talked before. We have a lot of data being generated with these sensors very quickly, but then analyzing them in a high accuracy will lead to performance and scalability challenges, energy efficiency, privacy, security, and cost. And what we want to basically promote in this workshop or like in this initiative overall is to have like a platform for exploring key computational challenges in health-related applications and see what we can do with our computer architecture knowledge and expertise to overcome some of these challenges to advance healthcare. And there are many different areas that we are interested in. I list some of them here, like computational biology. We talked about some of it today. neuroscience, wearable systems, medical robotics, mental health, medical imaging, agent-based simulations, and etc. This is, as I said, the list of topics that we covered in the previous iteration of this workshop at Micro 2025 in Seoul. And I again invite you to check our website and our YouTube link if you're interested in learning more. The next one will be happening in Sydney in HPCA, and the submission deadline for talk abstract is tomorrow. So if anyone is watching online, or also people in the room, you can consider submitting your abstracts, or also overall at any time if you're interested in the topic. It would be great to chat and yeah, to reach out to me, Konstantina, and Onur. And yeah, that's it. That brings me to the end of this lecture. And I think last lecture in the class. I hope that, huh? Oh, really? Ah, okay, cool. Yeah, so then let me add you also with the mic. So we will have some premiere lectures next week. New Year. Okay. New Year gifts. Yeah. Okay, yeah, thank you so much for listening. Are there any questions left from today? Okay, I guess we're done. Thanks.